quality systems. He has worked with a reputed institution, including NAVL Laboratories and ESIC Medical College, Kolkata, and currently serving as a microbiologist and infection control officer at Health World Hospital, Asansol. He is having extensive expertise in automated microbiology, culture reporting, ELISA, molecular diagnostics, and antimicrobial Steve Word program. His practical experience in infection control and clinician guidance makes him highly knowledgeable on the evolving challenges of the typhoid diagnostic and management in today's healthcare environment. So we are honored to have him with us today, and I'm sure the session will highly informative and insightful for all participants. So without further ado, I would like to hand over this platform Dr. Sanjeev Kumar. Sir, welcome. Thank you very much for joining us for this important and informative topic, typhoid. So, over to you, sir.
Thank you. I thank all of you to invite me to have a discussion on this very important topic of typhoid. So, basically, I will be more focused on the diagnosis of typhoid because I think most of us are laboratory-oriented persons. So let us have some basic knowledge of typhoid. First, what is enteric fever? Enteric fever is a systematic acute illness caused by Salmonella typhi and Salmonella typhi A, B, and C. So, Salmonella can be divided into typhoidal Salmonella and non-typhoidal Salmonella. So, if you think like what is the genus? Genus is Salmonella, and this genus Salmonella can be classified as typhoid Salmonella and non-typhoid Salmonella. The non-typhoid Salmonella have a different spectrum of disease. They cause gastroenteritis and septicemia, and they include many variants like Salmonella typhimurium, Salmonella enteritidis, and the typhoid Salmonella consists of Salmonella typhi and Salmonella paratyphi. And these Salmonella paratyphi are further of three types: A, B, and C. So Salmonella as a whole can be divided into typhoid and non-typhoid Salmonella, and the typhoid Salmonella can be divided into Salmonella typhi and paratyphi can be further divided into paratyphoid A, B, and C. And enteric fever is the fever caused by this Salmonella typhi plus Salmonella paratyphi A, B, C. So basically, there are, as far as the antigenic structure is concerned, basically there are three antigens: O antigen, H antigen, and VI antigen. O antigen is the somatic antigen, H antigen is the flagellar antigen, and VI antigen is the polysaccharide envelope or capsular antigen. O and H antigen are used in the Widal test, whereas VI antigen is poorly immunogenic, so it is not used for Widal test. Now come to the pathogenesis of typhoid. The route of transmission is fecal-oral. It spreads through the ingestion of contaminated food and water. After having ingested, it reaches our small intestine, where it penetrates the epithelial cells of the intestinal mucosa. From there, it reaches the submucosa, and in the submucosa, it is engulfed by the macrophages. Through these macrophages, they spread via the lymphatics to the mesenteric lymph nodes. And from the mesenteric lymph nodes, they enter the bloodstream to cause a transient bacteremia. So, fecal-oral route from ingestion to our gut into the small intestine. There, they invade the epithelial cells and reach the submucosa, and through macrophages, they enter the bloodstream to cause transient bacteremia. After the transient bacteremia, the bacteria has spread to different organs. They disseminate to the organs of the reticuloendothelial system, that is liver, spleen, lymph node, and bone marrow, and also to the other organs like gallbladder, and kidney, where they multiply. And the bacteremia, which is the clinical bacteremia, is actually the secondary bacteremia, which occurs from seeded organs. Means these organs, from these organs, they keep these organisms uh, seeded in the blood, and these seedling of the organisms from these organs causes secondary bacteremia and the clinical symptoms. Now the clinical feature, incubation period is typically 10 to 14 days. The fever pattern is very classic. It is a step ladder pattern, means every fever will have a spike which is more than the previous spike. Suppose today I'm getting a fever of 98.5. Tomorrow I will get a spike of 99.5. Next day I will get a spike of 100.5. Next day 101.5. Like this, every day the spike will be higher than the previous one, and then it will fall, but it will not touch the normal line. It will be remain at around 98.5, and then again it builds up to the higher level. So it's a step ladder pattern of remittent fever. As far as signs are concerned, those spots are usually seen on the trunk and chest, but it is seen in the Western population more than those in the Indian population, and the symptoms are very much common with the viral fever. It consists of headache, chill, malaise, cough, myalgia, arthralgia. Typhoid, there are early intestinal symptoms. So many patients complain abdominal pain, nausea, vomiting, anorexia. So these constitutional symptoms of headache, chill, cough, myalgia, arthralgia, along with the intestinal symptoms like the pain, abdomen, nausea, vomiting, and with that a classical step ladder pattern of fever, they signify that very likely the patient is having typhoid. Then there are two very important complications that can occur in a patient of typhoid if remain untreated. These are gastrointestinal bleeding and intestinal perforation. Both are very difficult to manage complications of typhoid. Now come to the epidemiological profile. Human is the only reservoir of the infection. The prevalence worldwide is 11 to 21 million per year. The mortality is 1.2 to 1.6 lakh death globally. The incidence of paratyphoid fever, the prevalence of paratyphoid fever is around 6 million cases per year. In India, there are more than 6 million cases of typhoid annually. In India, apart from typhoid, Salmonella paratyphi A has appeared to be increasingly reported in recent years. So in this map of the world, you can see those areas which are in dark red are actually the areas of the highest prevalence, and we in India are in the region of the highest prevalence of typhoid worldwide. Now the carrier state of enteric fever, there are two types of carriers, fecal carriers and urinary carriers. Fecal carriers, they shed typhoid fever, salmonella in their feces, and the urinary carriers, they shed the organisms in their urine. The carriers can be classified into temporary carriers or chronic carriers. Temporary carriers are those who excrete the bacteria for less than 3 months. The chronic carriers are those who excrete the bacteria for more than 1 year. And these chronic carriers usually have some abnormality of the gallbladder, like a gallstone, or they may have the abnormality of the urinary tract. As far as the prevalence is concerned, the carrier state occurs in 2 to 5% cases of the typhoid patients. And this persistent shedding of the organism by the carriers is a major source of infection spread. Now come to the laboratory diagnosis. So all of us know that in our basic teaching of the laboratory sciences, what we say, there is a rule called BASU rule. That means in the first week, the blood culture is the gold standard. In the second week, the antibody reactions like serology tests are useful. In third week, stool culture, and in fourth week, urine culture. These are the test of choice according to the duration of the fever. In the first week, blood culture, second week, serology, and third week and fourth week, stool and urine culture. When a patient of typhoid fever, suspected to have typhoid fever, comes in the first week, the choice of the test should always be blood culture and not the serology. Serology becomes positive only after first week of the fever. Now, culture isolation, blood culture, the first thing to remember is that it is very much productive in first week of fever. Second thing, whenever you are collecting the blood culture, at least 8 to 10 ml of blood per blood culture bottles, and minimum 2 such bottles, means total about 20 ml of blood should be collected. 8 to 10 ml of blood per culture bottle, and minimum 2 such bottles should be collected, and they are subjected to automated culture system like Bactalert. The manual system is very difficult to operate, means there are more chances of contamination, there are more chances of false negative results, but the automated culture system are highly accurate. So it is preferred to operate the blood culture through automated culture, and the specificity is 100%. Means if the blood culture is positive, it is sure to be a typhoid. And sensitivity is 40 to 60%. In the first week of fever, it is around 90%. But as the duration of fever increases, the sensitivity also decreases. Also, if the patient has already taken the antibiotics, the sensitivity will decrease. So overall sensitivity is 40 to 60%. Now the stool culture is used in third week. It is usually used to detect the carrier state. We can use the enrichment broth. We have to put the stool in the enrichment broth, like Selenite F broth. It is a very important point for the technicians who are working in the microbiology laboratory. If they are not using the enrichment broth, they will not get the growth. They will only get the growth of the normal GI tract flora. So you have to put the stool in the Selenite F broth, enrichment broth, and then from there you have to subculture it in the McConkey agar or XLD agar to get the growth. Now come to the Vidal test. It is basically a agglutination test. Nowadays there are both types of agglutination tests available: slide agglutination and tube agglutination. It measures the antibodies against H and O antigens of Serotyphi and Paratyphi. So what are the antigens used? H antigen of Serotyphi, Paratyphi A and Paratyphi B. So if you name them, H antigen and H antigen of Serotyphi is TH, H antigen of Paratyphi A is AH, and H antigen of Serotyphi B is BH. And then O antigen of Serotyphi, that is TO. What we do the serial dilution of the patient serum, and this is tested against the known bacterial suspensions. Titer is the highest dilution of sera when agglutination is visible. Say, you can see in this picture, the dilutions are like 1:20, 1:40, 1:80, 1:60, 1:320, 1:64, 640, and it is titer is the highest dilution of the sera where the agglutination is visible. So higher the titer means the higher the antibodies. So the higher dilution, suppose somebody is having a dilution of is positive till 1:40, and some other person is positive till 1:160. So the person who is having 1:160 dilution titer is having more significance than 1:40. Now come to the interpretation. The significant level of titer as suggested in India is of H antibodies is 1:200 and O antibody is 1:100. So at least this much titer should be there to call it significant, because there are local titers, because we all come to in contact with typhoid type of organisms in our daily means fecal habit. We sometimes in our life we all have consumed some contaminated water, due to which some antibodies are there inside our blood. So these low titer antibodies are not significant. Only a titer antibodies with which is more than 1:200 for H and 1:100 for O is significant. There are false positive and false negative results for Vidal test. The false negative test can appear in early stage. As I said, the Vidal test becomes positive only after first week of fever. So if you are testing in early stage, 3-4 days of fever, and you are testing Vidal, it will come negative. In late stage also, it may come negative. In carriers, the Vidal test may come negative. If there is prior antibiotic use, it may come negative. And if there is a lot of antibody inside the body, it may come negative due to projon phenomena. False positive, these are called anamnestic reactions. These are non-specific rise of antibodies due to unrelated fevers like malaria, dengue. Some patient is having malaria or dengue, and you get a rise in the titer of Vidal. So this is false positive. And if improper antigen reagents are used, if there are flagellar antigen inside the reagent, it may give false positive reaction. So there are multiple false negative and false positive means cases as far as the Vidal test is concerned, and there is a chance that low level titer will be present in the endemic population. So a fourfold rise in antibody titer by testing paired sera after 1 week interval is more meaningful than the testing a single titer. Suppose somebody is having 1:1, 1:80 O titer, TO titer, and you are in confusion whether this titer is significant or not. You may repeat the test after 1 week, and if there is a fourfold rise, means 1:180, twofold rise is 1:160, fourfold rise is 1:320. So if the titer goes from 1:80 to 1:320 in 1 week, it is much more significant than getting a single titer because of a lot of false positive and false negative possibilities. Now, let us interpret the report of a Vidal test. What are the reports which may appear with you, and what is its interpretation? So before knowing that, you should know that O antibodies appear early and indicates recent infection. So O antibody appear early and disappears early. So presence of O antigen means there is recent infection. H antibodies appear late and disappear late. So they represent convalescent stage. If the H antibody is without O antibody, it means the infection is delayed infection, it is in a convalescent stage. Second point to be noted is O antibodies are serotype non-specific. That means they range in all the infections, S type Phi, paratype Phi A, paratype Phi B. So no matter what infection it is, the O antibody will rise, right? But the H antibodies are specific. So having this much understanding in mind, let us interpret the results of typhoid. So TO is increased, TH is increased. That means it's a typhoid infection. TO is increased. AH means paratyphi AH antibody has increased. That means it's a salmonella paratyphi A infection. TO rise, BH rise. It is a salmonella paratyphi B infection. Only TH rise. There is no rise in TO. That means it is either a convalescent stage, the recent stage of typhoid fever has passed, it is a convalescent stage, or it's a anamnestic response. Only TO has increased, but TH has not yet increased. It can occur in recent infection because TH appears late. So it may occur in recent infection that only you are seeing only TO rise. And if there is rise in all TH, AH, and BH, it signifies post-vaccination stage. This type of feature appears in post-vaccination stage. The other antibody detection system, there are many other antibody detection systems for typhoid. Like one is very common, typhoid test. It utilizes the OMP antigen and detects both IgM and IgG separately. The limitations are in early infections and some secondary infections, detectable level of IgM antibodies may be low. Some patients may not produce detectable level of antibody within the first 7 to 10 days after infection. So, like Vidal, there may be, if you are doing this test in 3-4 days of fever, they will remain negative. It may remain negative up to 7 to 10 days of fever. And high titer of rheumatoid arthritis antibody, SLE antibody may show cross-reactivity, means they may come false positive in case of rheumatoid arthritis antibody and SLE antibody predicting the blood of the. There are nested PCRs to detect difference and differentiate salmonella by targeting genes like flagin gene, IROB gene, FLIC gene. They are highly sensitive and specific. So why are these nested PCR highly sensitive and specific? Because they are nested PCR. What do you mean by nested PCR? Nested PCR, in nested PCR, what we do, suppose there is a one DNA strand and this we have put primers to identify a target sequence in that DNA strand, and then it will replicate, it will do a PCR. Again, this target, when the PCR is done, the next target sequence will attach, the next set of primers will attach with each amplified DNA products. Means the second set of DNA will hybridize with the second primers. So two primers at different level we are using to detect one organism. So this gives a very high sensitivity and specificity to the assay. So nested PCR we are using for detection of typhoid. Now I will focus on most recent advancements in the detection of typhoid as well as in overall microbiology, because the methods I will be talking about in my next slides are the future of microbiology. They are used for the detection of typhoid also, and they are used as such in the microbiology field for as a pipeline projects which will replace the current system with their advancements. So the first is MALDI-TOF MS. What is MALDI-TOF? MALDI-TOF stands for Matrix Assisted Laser Desorption Ionization Time of Flight Mass Spectrometry. What is this? It's a high-tech mass spectrometer machine. You can see the machine used to quickly identify bacteria by analyzing their protein. So mind that this is not a PCR system. It do not identify the DNA. Rather, it identifies the protein. How it is done? It is done by weight-based sorting. Every bacteria has a unique combination of proteins. The machine turns these proteins into electrical mist and shoots them down a tube. Because lighter proteins fly faster than heavy ones, they land at different times, creating a unique speed profile or fingerprint that names the bacteria. I will explain it. Okay, how do we do it? Just pick a grown bacterial colony from a culture plate. So we had inoculated the sample on a culture plate, and when the bacterial colony grows up, we will pick the grown bacterial colony. Then we will smear it on a metal plate. This is the metal plate. We will smear it on the metal plate, and we will add the special chemical matrix. We will add the chemicals, and we will insert this plate into the machine. Right now, a laser. See, this is the laser. This laser will shoot into the machine, shoot it, right? And this shooting by the laser will cause vaporization of these protein matrix, and these vapors will be allowed to go through a tube, and they will go through this tube. Like this laser it is applied on the plate, and this will vaporize the protein, and these proteins will travel through this tube, right? And they will travel through this tube with a particular speed. So the proteins will be divided into its components, and the component which is lighter will travel faster through the tube, and the component which is heavier will travel slower, and they will hit the detection system, and the detection system will analyze how the proteins are hitting the system. So the detection system finally, when all the proteins hit the system, all the lighter and the heavier proteins in different time matrix have reached the system will analyze the pattern of the proteins, and with this analysis, it will find out which bacteria it is. So this is a highly complex system, but this is actually available nowadays in many parts of, I know in Kolkata, at many places, it is now available, and this is the future of detection of organisms. Now, second method is whole genome sequencing. What is whole genome sequencing? It's a comprehensive genetic test that reads the entire DNA blueprint of the pathogen, right? So the entire DNA blueprint will be read in this system. How it is done? It is done by letter reading. It extracts the entire DNA strand from the bacteria and reads all its genetic code, A, T, C, G, everything from top to bottom. It will read, and by reading the whole book, it identifies the exact strain and every single drug resistance these organisms carry. So I'll make it more simpler. So how it is done? Again, pick a grown bacterial colony from the culture plate, just like in Mitra. Break open the grown bacteria to extract its pure DNA. So we will do the extraction process. Then the DNA are cut into pieces and are tagged, and they are fed into a machine called sequencer machine, and then the computer software is run to reconstruct it and analyze the entire genome. So this looks a bit difficult, but I will make it easy for you. Suppose you want to read a book where only 4 letters are written, A, T, C, G, in different sequence, A, T, C, G, G, C, A, so A, T, T, everything is written, but you don't know how to read it, right? So what you do, you take a scanner, but a scanner has a capacity to read just one sentence at a time. So in order to make it read, you will you will break the whole book into different sentences, different small sentences. So if suppose one sentence is, uh, Ram goes to the market in the morning, and the scanner can read only two words at a time. So you will cut it. Ram goes one part, to the one part, market in one part, and the morning another part. So there are four parts. Then you will feed them into the scanner, and the scanner will read it. But when it is reading, what is it doing? It is reading. You don't know which part has come first and which part has come later. So when you are reading, what you are doing, to the market, it has read to the market, then Ram, then in the morning. So in this sequence, the scanner is reading. So it makes no sense. So you will feed it in a software which will analyze it, what it has actually read, and it will, by analysis, it will reconstruct the sentence that Ram goes to the market in the morning. And it will make the complete sentence, and now it is meaningful. So this is the same thing. The bacterial DNA is cut into pieces, and then it is tagged, and then the sequencer will read everything, and then these broken sequences are reconstructed, and the whole genome is analyzed. So this is the process of whole genome sequencing. The third process is CRISPR. CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. It's a modern molecular tool that uses programmable search guide to find a specific typhoidal DNA. How it is done? It uses a customized genetic gRNA. So there is gRNA, like mRNA, RNA, there is gRNA. And RNA can combine with a DNA, we all know. So it uses gRNA, and this gRNA will have attached molecule. This molecule is CRISPR enzyme. So there is a system of a gRNA which will guide RNA, which will attach with the target DNA sequence of the organism, and this gRNA is attached with a CRISPR enzyme, right? And whenever this gRNA finds its counterpart, if the typhoid DNA is present, the gRNA will attach with this typhoid DNA, and this attachment of gRNA with the typhoid DNA will activate this CRISPR enzyme. And CRISPR enzyme, what it will do, it will chop the nearby reporter dye and will cause a visible color change. So what's happening? In the sample, you are adding gRNA, which is attached with the CRISPR dye. And along with that, you are also adding some reporter dye. This gRNA, when it attaches with the genome, with the DNA of the typhoid bacteria, it will activate this attachment will activate the CRISPR enzyme, and this CRISPR enzyme will cut the reporter dye, and the reporter dye will give the color. So the method is extract and slightly amplify, extract and slightly amplify the target DNA from blood and urine. This point is very important, that we have to slightly amplify. If we do not amplify the target DNA, what will happen? The dye, the color of the dye, which gives the positive signal, will be very low in intensity. Then after extraction and amplification, the CRISPR enzyme with the guide RNA will be added. If typhoid is present, the CRISPR enzyme system locks on and cuts the reporter dye, and read the visual color change like a pregnancy test of fluorescence. So the visual color change can be read like a pregnancy test, or it may be read like a fluorescence signal. So now the advantages, disadvantages, and utility of all these three new technologies. Regarding multi-top, the advantage is it takes less than 15 minutes once the bacteria is grown. So when you put it in a multi-top machine, only in 50 minutes, 15 minutes, you will get the results. The cost is very less in materials after buying the machine. Mind it, after buying the machine is very costly, but the cost of identification is very less. Whole genome sequencing, it tells you everything, the exact strain, because it is using the whole genome, so it will tell you everything, the exact strain, the geographical origin, the evolution, everything it will tell. Along with the full resistance profile, it identifies every single antibiotic resistance genes automatically. And CRISPR, the advantage is it can be done bedside or in the field of clinics. It's a point of care test, just like immunochromatography. It's also a point of care test. And after evolution, it will be a point of care test, just like we are using all the immunochromatography tests, which are based on the antigen antibody reaction. It will be the next generation immunochromatography test, which will based on the DNA RNA hybridization and CRISPR technique. There is no expensive machinery involved. The results can often be read with the naked eye via a simple color change. It bypasses the need for expensive thermal cycler. So in all of the PCR machines, the machines which are identifying the DNA, what we require is a thermal cycler. The temperature will have to go to 90 to 95 degrees C, then again come back to 50 to 55, then again go back to 75 degree, and this change in the temperature keep on going to make the PCR happen. But here this thermal cycling is not required. Though it is a DNA RNA hybridization based system, the thermal cycling is not required in this CRISPR system. And it has a culture independent potential. For both multi-top and whole genome sequencing, the first step is to grow the organism in culture, which will itself take 24 to 48 hours. But CRISPR is a culture independent mechanism. It can detect the bug directly from the blood without waiting days for it to grow. Now the disadvantages. Multi-top MS, it requires culture. We still have to wait for 24 to 48 hours to grow the bacteria first. The machine itself is incredibly expensive. And multi-top is excellent at identifying the genus species. For Salmonella, it will definitely identify up to Salmonella enterica, but it struggles with the subtypes like typhoid versus paratyphoid. Nowadays, the new system also detects typhoid versus paratyphoid, but the older multi-tops, they are still struggling to further classify Salmonella enterica into typhoid versus paratyphoid. Whole genome sequencing, the disadvantages are just like multi-top, it cannot be done directly on raw sample. You must wait 24 to 48 hours to grow a pure bacterial colony first. It is high cost. The test kits and sequencing consumables are very expensive. The sequencing process, the software is very slow process. It takes another 1 to 2 days. And the bioinformatics, the process of rearranging to get a meaningful sequence, it requires a computer complex software, and it requires data experts to read the results. CRISPR, the disadvantage is it's still in a developmental phase. It's a new technology, and it is still in developmental phase. It has not come to the market yet. And pre-amplification needed, as I said. If you do not do a pre-amplification, the signals will be very weak. So usually require an initial isothermal step like loop-mediated isothermal amplification lamp test to make the signal strong enough for paper strips. And these enzymes and RNA guide molecules require a strict temperature controlled supply chain. If this supply chain is not maintained to take this technology to the ground level, you will get false positive or false negative results. The utility method of MS, it is used as the first line daily routine tool in modern hospital lab. As I said, many modern hospital labs have adapted multi-dose MS already. They immediately confirm if the grown species is in blood culture is someone typhoid or not. Whole genome sequencing its utility is in the public health agencies and research laboratory to track typhoid outbreak, trace the source of contamination, and monitor global drug resistance. And CRISPR, it is ideal for low resource areas, rural field clinics, and rapid screening, where sophisticated laboratory infrastructure does not exist. So these are the three methods I wanted to tell you, which are now being used or will be used for the diagnosis of typhoid and other organisms in the microbiology. Now come to the drug resistance. Earlier, the first type of drug resistance which appeared in typhoid salmonella, it appeared in 1989. This was the first MDR salmonella, which is resistant to chloramphenicol, ampicillin, and cotrimoxazole. Then after the emergence of these MDR strains, people selected ciprofloxacin because ciprofloxacin were sensitive and it became the drug of choice. But nowadays, ciprofloxacin resistance has become increasingly common. So nowadays, about 70% of the strains are even ciprofloxacin resistance. After that, the treatment shifted from ciprofloxacin to ceftriaxone, and gradually the ceftriaxone, the resistance have started appearing in ceftriaxone also, and now till today, the ciprofloxacin resistance is less than 1%. So still we can very safely use ceftriaxone for the treatment of typhoid. So based on this, the treatment guidelines say the first line of therapy is ceftriaxone, 1 to 2 gram IV daily for 10 to 14 days. The alternative is azithromycin, 1 gram per day orally for 5 days. Now, last, the prophylaxis and vaccination. First thing is the gender prophylaxis, the most important part for prophylaxis to control cases and carriers. And how can we control the cases? First, we have to know the cases and carriers. So first we have to detect the cases and carriers. And after detection, we have to properly treat them so that they do not spread the organism in the community. So here also, as a step of prophylaxis, the detection part is very important. Then sanitation, purified water, hand washing protocols, and food hygiene is very important. And third is vaccination. Vaccination provides short-term protection. It is indicated in travelers going to endemic areas, people attending mail-outs, and household contacts. So basically, there are two types of vaccines available. VI, capsular polysaccharide vaccine. It's a parental vaccine which can be given IM or subcutaneous, and there is a oral vaccine called Typhoid or Ty21A, which is a oral live attenuated vaccine. So with this, I conclude. Thank you.
Thank you very much, sir, and your valuable time and sharing such an insightful session on this typhoid in the modern era. So this session was extremely informative and engaging, and we sincerely appreciate your contribution and look forward to many more academic interaction in future. Thank you once again for making this webinar very wonderful and informative. So, thank you very much, everyone, for joining today's insightful webinar on typhoid. So, we will meet you again in the next webinar in the coming days. Thank you very much, all
Sir,
of you.
sir,
Thank
we
you
will
very much,
take
sir. Dinu, thank you.
So there
Questions
is
are
some
there?
yes, sir. Some questions are asked. We'd like to discuss those
You
questions.
can go ahead. Yeah.
Thank you, sir, Dr. Sanjeev. Thank you for this insightful session on this typhoid. As a tropical country, as you already mentioned, that in India there is a real burden, and in eastern part of India this is really endemic, especially in West Bengal. As you said, sir, that in this particular case of typhoid bacteria, so there are carrier stages, and it goes to
Right.
a carrier stage. So one of our participants had asked a question on this particular stage, that in carrier stages, as the bacteria resides over a longer period of time, so the IgG antibodies and little IgM antibodies will be present. So how to differentiate the acute cases and chronic cases as a diagnostician?
IgM cases will go away, na? IgM is for recent infection, and carrier stage are usually identified by stool culture. So culture is better method for the detection of the carrier. Use stool and urinary carriers using culture.
Thank you, sir.
And
Uh,
there are and VI antibodies which are utilized for the detection of the carrier stage.
The next question is it has been noticed that during monsoon time, specifically during high dengue prevailing cases, this typhoid is also coming positive. So, how do you report these type of cases? Like, sir, as you already mentioned in your presentation that it has some related unrelated disease like malaria and dengue cases. They are the false rise of the non-specific rise of the IgM antibody occurs. But as a reporting microbiologist, how do you report these cases? Because this is very common in this monsoon time, specifically each dengue positive and the typhoid same time it comes positive.
No, for that, I think we should rely more on the blood culture. Blood culture is very highly specific, and if we adopt to sending samples in the first week of fever, it is. I have seen it is very highly reliable. I have given you also cases na, where blood culture was positive and some other dengue and all were positive, and we concluded out that yes, typhoid is the actual reason, cause of reason. So blood culture should be more relied on.
I mean, reporting part, we will report dengue positive as well as typhoid positive, and
Right,
then
right,
with a then with a note that to as a confirmatory test we should do the blood culture to confirm the typhoid case.
right.
Thank you. And sir, one thing is another question is that once there is no symptom of typhoid, but this rapid card is positive, but the Vidal is coming negative. How do you handle these cases?
Okay, so I want to tell you a story about this. My father-in-law,
Yes, sir.
had, he was in Patna and I was in Malda that time. So he had fever and his this rapid card was positive. Typhoid was positive, and he was given ceftriaxone, and he was taking ceftriaxone for last 10 days or 14 days. I also said, okay, you go on taking you complete the course, and then nothing happened. He was continuously having that fever. So I called him to Malda, and I again rechecked it with the typhoid test card. And this time I found this is negative. So I don't know what had happened, but what was happening, the fever was never touching more than 101. The fever was around 99 to 101. The PSR was around 90, 90 to 100. And there was some neck pain associated. So I consulted medicine experts, and they said they also could not find out why this is happening. They said, okay, they neck pain, you go and do some yeah, ultrasound of the neck. And what came out was a thyroiditis. It was a subacute thyroiditis, right? And we were because of this false positive result. And he was given steroids, and he responded very well. And this gave me a conclusion that there are false positive tests, and we should always look for the other clues which may be hidden in, suppose, high ESR. Why the typhoid, a patient with typhoid will have a ESR of 90 or 100? Why not his fever will touch more than 101? Why will it remain under 101 always? So these clinical findings are very important whenever you are deciding. Don't decide one. That's why we right now you correlate with the clinical findings. So decisions should be made along with the clinical findings, not only based on the tests.
Yes, sir. Correctly said, sir. Like we generally correlate CBC also. The
Right.
total count seems to be very low. But in case of dengue, as the attendee has asked, in both dengue and typhoid, at both I mean, for the both the diseases, this total count goes down. But yes, not platelet definitely.
Hmm.
So the correlation between different departments of the lab is very important.
And the correlation with the clinical findings is also very important.
Yes, correct.
The GI
Nowadays,
symptoms, the GI symptoms which I said, the GI symptoms are very frequent in typhoid
yes, correct.
rather than
This part
dengue.
this part is correct, sir. That but nowadays what happens? You know the maximum the culture of referral system or referral labs are here. So they send the samples, but there are no clinical this
True.
information is shared with that particular sample. So it's very difficult to interpret the report at a referral part as well.
Very correct.
Yes. And sir, two more questions we'll ask. Someone asked that in the market we have seen many ICT cards available, but due to the differences in the target molecule, differences are noticed in final report. Some shows negative while some are positive. As a lab technician, it's very difficult to report how you are handling such cases, and is there any other specific criteria you follow to report those discrepancies?
The cards which are using the OMP, na, they are more sensitive than the laboratory separate cards. So maybe that may be the thing. But whenever in problem, you should cross check with a means different uh, type of system before reporting.
So, do you think that we should continue with this two Vidal part, and as a slide?
Q-Vidal is actually uh, more means, sensitive than the card test or the slide Vidal test. And but nowadays, actually, what happens now? The tube Vidal, nobody, no clinician want this test to be reported one day after, because tube Vidal will take one day, and everybody want the rapid results. So tube Vidal, that's why tube Vidal is out of fashion right now. It is only done in the medical college type of laboratories or government setups. In private
Research
setup,
setups.
everybody is having.
Correct, sir. Correct. But as a clinician or as a practitioner, we should focus on these that which were conventional, which were taking time, but it was more reliable than those rapids.
Actually, what I do when I get some test which is means say positive or even low positive by card test, I tend to check it through Vidal test, right? And if the Vidal test is positive, then I can be more confident in reporting the results, because Vidal test reagents are not that costly. If you cross check it with say in case of positive results, you if and particularly if the O antigens are having some agglutination, it's very highly means there is high chance that this is a positive result because O is more specific than the H antigen. So at least check the O antigen whenever you are doing some card test. My recommendation would be like whenever you are doing the card test and you are getting a positive result or a false or a low positive result, you can cross check with your viral test reagent, uh, particularly the O antibody. And after cross checking, if you give the result, you will be more confident.
Yes, thank you. And one last question is Mr. Ganesh Meher has asked a question that how is the sensitivity and specificity of CRISPR in diagnosis of typhoid?
The sensitivity is very high. That's why CRISPR is being developed. And but the problem is that this CRISPR technology requires a amplification before installation of the CRISPR molecules. So this, if we do not amplify the DNA, the results will be very faint, and we may not be able to read the results. The color changes or the fluorescence emission will be very faint. So this particular problem, the scientists are focusing on because first doing a loop mediated amplification and then doing the CRISPR, it do not fulfill the criteria why this test they want to introduce this test. The criteria, the purpose of introducing this CRISPR test is that it should be used at a point of care test with minimum level of machinery or expertise involvement. And if this test uh, if this test requires an initial level of PCR sort of thing, this the purpose is not fulfilled. So the scientists are working on it. But if when this problem is resolved and when it will come in market, it will definitely my gut feeling is that it will definitely replace all the antigen antibody based tests, and the CRISPR will be the next uh, model of the immunochromatography test and with a highly high sensitivity and specificity.
Correct, sir. Rightly said that molecular is the future of diagnosis. That we have seen in case of this HIV, HBC, and HCV also. NAT testing is being promoted quite rapidly in last decade. So yes, correct that molecular diagnosis in coming days will be evolving in India, particularly in India. Thank you, sir. Thank you for your time. And all questions were really asked, and I truly hope that all our participants get something benefited from this uh, insightful presentation on typhoid, which is a very common in India.
I wanted to actually uh, emphasize on the interpretation of the Vidal test what they are doing, because there are many cases where O antigen is negative, but H antigen is positive, and the doctors keep on writing antibiotics based on those things. So these things should be checked. Means merely the writing the H antibody without any O antibody, it most of the times it is just a anamnestic reaction or a convalescent stage, and we should and whenever we are giving the test for I mean, giving the blood for any test like blood culture or the Vidal, what we are doing, we are taking antibiotics for 7 days and then we are giving the Vidal test. How can it be positive? Or for blood culture, there is a requirement of writing more of blood culture in first week of fever. As I am in an acute care setting, so whenever patient comes with fever, as a protocol we take to blood culture. That's why I get a lot of STFA. I have discussed with you earlier also. We get a lot of stamina typhi because we are able to take the blood in the first week of fever. But when you go in community level, standalone OPD based labs, you will not get these blood cultures positive because the doctor first write antibiotics. After 7 days of antibiotics, then they still rely on Vidal test. So these things decreases the chance probability of getting the accurate results of the tests. So we should first try to be very cautious about what is the period of the illness, what is the period after, how many days of fever has gone, and we should write the test accordingly. And second thing, whenever a test result comes, we should be very cautious in interpreting the test results, because there are cross reactions and there are multiple interpretations of viral test. And even in immunochromatographic test, there are cross reactions, dengue coming positive and Typhoid coming positive. So we should cross check with other tests also, and then come to final diagnosis. And very important, we should try to correlate everything with the clinical findings, because right, I said the clinical findings are very means peculiar for typhoid. And if you look at some other things which comes with the fever, their clinical presentation will be quite different from the clinical presentation of the typhoid. So based on that also, you can actually identify which test is correct if two tests are coming positive simultaneously.
Yes, sir. Correctly said. Thank you, sir. Thank you so much
Thank
for
you
your time.
very much, sir. So thank you very much,
Yeah, looking forward
Dinabandhu, for
to
raising
more.
the very valuable questions. And yeah, and sir, once again, thank you very much for taking out your precious time for this informative webinar. Thank you very much, sir. Thank
Thank
you.
you.
Thank you so much, sir.